Now that we know the digestive anatomy of the human body, let's go ahead and talk about how food is digested. That means that chemical bonds are broken. Now, we already talked about the macromolecules, carbohydrates, lipids, and proteins. We're not going to deal with nucleic acids. And we already mentioned that there are some things that are not going to be digested. For example, cellulose. We could also say lignin, Lignin, is also not digested. Lignin is basically what wood is made of, and you need an enzyme called ligninase, which is only found in fungi and bacteria. So that's not going to be digested either. But let's take a look at the three macromolecules that I mentioned. So we've already discussed that they're eventually going to go down to amino acids, fatty acids, and simple sugars. We started out in the oral cavity, and I mentioned that saliva contains amylase, which breaks down carbohydrates. An example of that, so let's have an example here. We don't want that. So for example, a carbohydrate starch would get broken down into a disaccharide maltose. So you need to understand that in the oral cavity we're breaking down carbohydrates, but not all the way to simple sugars. We're going to disaccharides. They'll be broken down later on. So it's really a pre-processing. Then we go down into the stomach. Now the physical sensation of food entering the stomach causes the stomach to distend, D-I-S-T-E-N-D. That's a good word to know. Let's go ahead and write that one down there. Distend. That means to get bigger. And that tells the stomach that food is in there. Also, the nervous system is alerted by simply the sight and smell of food. So both those things cause the production of a hormone, and that hormone is called gastrin. Let's write that down there. And gastrin is secreted by the stomach itself from the stomach lining. And also from the stomach lining, there's two types of cells there, parietal and chief. You can see right here. And they secrete hydrochloric acid, and they secrete pepsin. But they don't secrete pepsin in that form. They actually secrete it as pepsinogen. Now, when you see something with the GEN on an enzyme, that means that it's not really active yet. And the reasons why it's not active is because if you had active pepsin running around the stomach, it would start to digest the stomach itself, and that's no good. So it's stored as an inactive form, and then hydrochloric acid comes along and activates it and turns it into pepsin. And pepsin goes ahead and breaks down proteins. And once again, it doesn't break down proteins to the amino acids. It breaks it down probably into dipeptides or tripeptides. And later on, those will be broken down into the smaller amino acids. Then we come down to the small intestine. Now, the small intestine is where digestion is really going to take place, and the reason why that happens is that it's actually connected to an accessory organ, not part of the GI tract, but something which is along the side called the pancreas. And the pancreas is connected to the small intestine by something called the pancreatic duct. And the pancreas produces a ton of enzyme, too many for us to really write down here. But all those enzymes run down the pancreatic duct and are emptied into the small intestine. Among those enzymes are amylase, who we met before in the oral cavity, so we're going to get more amylase. We're going to get trypsin. That's another big enzyme that will digest proteins. We're also going to get lipase. That's somebody who digests lipids, and lots of other enzymes. Not only that, but the pancreatic juice, which contains all these enzymes, also contains bicarbonate. Now remember, bicarbonate from your chemistry class was a base. And the reason why it's got to secrete that is that will neutralize all the hydrochloric acid that's still coming down from the stomach. So tons of these enzymes, what we call pancreatic juice, or what they actually call a cocktail, comes down from the pancreas into the small intestine and does the majority of the digestion. Somebody else who's coming down there. The liver, which is another accessory organ, produces something called bile. And bile is actually stored in the gallbladder. Not produced in the gallbladder, but that's when it's stored. And the gallbladder is also connected to the small intestine by something called the cystic duct. And it sends all its bile down into the small intestine, right? Now, what does bile do? Bile does something called emulsification. It emulsifies lipids. What does that mean? It means essentially it dissolves the lipids in water. So remember from your chemistry again, fats do not dissolve in water. So if they don't dissolve in water, how can all those enzymes who are dissolved in water digest fat? They can't. So you have to add something called bile, which emulsifies. It forces the lipids, the fats, to dissolve in water, and then all the enzymes can work on it. So that's really, really important for the digestion of fat. Okay, now, everybody at the small intestine is starting to get digested pretty good, but there's actually one more place that major digestion takes place. And that is actually in the lining of the small intestine itself, or what we call the brush border. Now remember from your basic biology last year, we talked about the fluid mosaic model. And we talked about how membranes actually contain bunches of proteins in them. And a lot of those proteins are, surprise, surprise, they're actually enzymes. There's tons of enzymes, therefore, in the wall of the small intestine, the brush border. Among them are these enzymes: lactase, sucrase, maltase, lipase, nuclease. And what these guys do is they actually do the final digestion. So, for example, suppose in the small intestine you had sucrose. It hits the wall of the small intestine, and an enzyme in the wall called sucrase would actually then dissolve it into the final fructose and glucose simple sugars. So, bottom line, when things hit those enzymes in the small intestine wall, they finally get converted into amino acids or simple sugars or fatty acids. Now, why would the final enzymes be in the wall of the small intestine? Because, remember, the small intestine is going to actually absorb those goodies into the bloodstream. And all these enzymes are actually contained in the wall of the small intestine in a bunch of folds. These folds kind of look like that, like fingers almost. The reason why we have that is to increase surface area. And those folds, those fingers, are called villi. So these guys contain all these little enzymes here, right? I'm not drawing a very good picture. And as your macromolecules hit them, they're finally digested, and then they enter into the bloodstream, and off they go. We'll draw a better picture on this tomorrow when we discuss in more detail what the villi are.